Hey, my name is Peter Risa. I'm a first-year medical student here at LSUHSC in New Orleans. Foremost, I want to thank Dr. Fern Chen and her team for allowing us this opportunity to present. And secondly, the judges for making this opportunity possible. So I joined the Marrero lab over the summer, where a portion of us were studying osteoarthritis. Now, at the time when I joined, Dr. Marrero was actually proposing a translational study, which was to repurpose an FDA-approved drug, minoxidil, and look at its possibility of serving as an antifibrotic therapeutic. The title of our poster is Attenuating Collagen Deposition by Synoviocytes from Osteoarthritic Patients with Synovial Fibrosis. Here's a large-scale view of our poster. Now, osteoarthritis is a common disease and is characterized by the degradation of articular cartilage. And articular cartilage is the cartilage that lines the two bones. And so it's typically defined as a wear and tear disease. Now, this wear and tear disease, is a hallmark of it is fibrosis. And fibrosis is just the deposition of extracellular matrix, primarily collagen 1. Now, fibrosis is largely driven by TGF-beta, transforming growth factor, and its whole cascade. And so this stimulates a couple of things, such as smooth muscle actin, myofibroblast differentiation. And myofibroblasts are the ones that actually deposit 95% of the collagen. And so this is important. So that's going to increase collagen 1 synthesis. Now, collagen 1 is this molecule you see over here on the right. These inter, and that's very important, bonds, intercovalent bonds, are pyridinoline cross linkages. And we're just going to call them PYD for short because pyridinoline is too tough to say. So these pyridinoline or PYD cross linkages are actually what are proposed to make collagen tough. We have some preliminary data, and we showed that, in patients with high fibrosis, PYD cross linkages, or the enzyme that facilitates PYD cross linkages, is actually elevated, and that enzyme is LH2B, or lysyl hydroxylase. Recently there was this drug minoxidil, which you probably know as a therapeutic for alopecia or hair balding, has been repurposed within the last 3 or 4 years as an antifibrotic, specifically in clubfoot patients as well as idiopathic pulmonary fibrosis. And so they were able to show over a span of 21 days and actually 8 days that minoxidil was able to decrease the activity of LH2B. And so that is LH2B is that lysyl hydroxylase, the enzyme that facilitates this cross linkages. And so in that way, minoxidil could be, making weaker collagen. And that way, the sturdiness and that sturdiness is what causes the difficulty in osteoarthritic patients and the inflammation as well. And so minoxidil became a proposed therapeutic. And one thing to note is that minoxidil actually works intracellularly. So not only is it FDA approved and it's already a safe therapeutic, it actually works intracellularly, meaning it won't work on collagen that's already deposited in the extracellular matrix. And therefore, it's rather safe. You don't want to disrupt that extracellular collagen. And so with that, what we decided to look at was minoxidil's potential as an antifibrotic agent, applying it to the synoviocytes and synoviocytes extracted from osteoarthritis patients. And so we're going to look at the PYD count, right, the cross linkages, as well as the total collagen deposition. Now, here are our methods. This is a nice figure. So Dr. Daza and Dr. Marrero are co-directors of the LIM, the LSU Integrated Musculoskeletal Bank. And in this bank, they actually just collect all the tissues that are extracted and not needed from a TKA, a total knee arthroplasty. And so that includes, the synovial fluid, the meniscus, like the tibial plateau, just bones and extra ligaments that are not needed. So what we were able to do is we were able to take some tissue, fix and process it, stain it for LH2B, which is that enzyme that facilitates the cross linkages, and then look at it for confocal microscopy, look at it under confocal microscopy. Second, we were able to culture some cells, so some synoviocytes, fibroblastic synoviocytes, transform them with TGF-beta1 for 48 hours, stain them, and then go ahead and look at smooth muscle actin and collagen 1. And so you'll be able to see that here in just a moment. Those images will be over in the results section. Finally, we were able to look at minoxidil and its potential as an antitherapeutic, as an antifibrotic, as a therapeutic. And so we treated with minoxidil for 8 days. And so with that, we were able to do our cell lysate extraction, extract our RNA and our protein. Our RNA, we're going to be looking at a qPCR and actually look at the transcription levels of LH2B. That's a little later. It was a summer project, so we focused on the proteins. And with the proteins, we ran ELISAs against PYD and collagen 1. And finally, we analyzed this data in PRISM. Here are our results. And so if you could look at the figure 3 on the left, we can see that going from A to B to C to D, we have increasing concentrations of TGF-beta1. Now, TGF-beta1 was this master profibrotic compound. And so we can see that in green, we have collagen 1, and in red, we have smooth muscle actin. And as we move from decreasing concentration to increasing concentration, we can see more green, so more collagen deposition, as well as more red, which is smooth muscle actin, which is myofibroblast differentiation. So we were able to, see pretty nicely under confocal microscopy that TGF-beta is actually a profibrotic agent, and we were able to show that. In figure 4 here on the right, we were able to show that LH2B expression, and this was that enzyme, that protein that facilitated cross-linkages, is actually higher in highly fibrotic patients. And so highly fibrotic patients, and the PRO's understanding is that these highly fibrotic patients would have more PYD cross-linkages and thus sturdier collagen, correct? And so we can see that here that it was actually statistically significant when you subcategorize them by high fibrosis and low fibrosis. And so that was a very interesting finding. And so that's a very nice potential for minoxidil to come in, which actually works on LH2B transcription. So here are our results with minoxidil. And so on the left, if I can orient you, we have total collagen 1 deposition, and we have here on the x-axis no treatment and minoxidil treatment. Now, when we treated with our cells, when we compared our cells between no treatment and minoxidil treatment, we were able to see that there was similar collagen 1 total deposition. And so this is actually an interesting finding. This does not, this bolsters the current literature and actually is sort of what we were hypothesizing, surely what we were hypothesizing. But as we turn our attention to the right, we have the total PYD linkages and this cross-link count. Now, what we were able to find was that there was no significant difference or no significant decrease in PYD cross-linkages when comparing the minoxidil treatment group to the no treatment group. And so this was, stating or suggesting that minoxidil really has no effect on the total number of PYD cross-linkages. Now, these findings are summarized here really nicely, and I'm going to go ahead and just use the images. Really briefly, we can see that TGF-beta is a profibrotic agent, and it's able to increase the green, which is collagen deposition, as well as the red, which is smooth muscle actin. Over here, we will show that LH2B expression is increased in highly fibrotic patients, and thus that's the understanding that is the reason why they have sturdier collagen. We were able to show that total collagen 1 deposition does not decrease or does not increase actually with minoxidil treatment, which is sort of what we proposed and we actually hypothesized and expected because it works intracellularly. And then PYD actually was not able to elicit an effect, or minoxidil was not able to elicit an effect on PYD total cross linkages. And so here's our discussion, and we were able to see that total collagen was not changed, but the pyridinoline PYD cross linkages was changed. And it's important to note that LH2B expression did change, and we showed that, but that was because we were able to group and subgroup by fibrotic patients. And so when we look back here, this is actually aggregate data, right? So we only have 9 patients here, but this is the aggregate data where we read a subgroup, and because it's a blind study, I'm not able to yet, where we read a subgroup by, fibrosis and high fibrosis patients, potentially we could see a difference. Over this, at the start of this year, early in January, Dr. Marrero was able to publish a paper looking at osteoarthritis with comparison to race. And he showed that, race had an effect and even sex had an effect with osteoarthritis. And so if you're able to subgroup by several things, the potential is there for minoxidil to elicit an effect. But at the moment, our pilot data really suggests that there is no difference in collagen 1 synthesis or post-translational PYD cross-linking. And so the next step would be to go ahead and emulate the experiment with a larger sample size, go ahead and then subgroup this data over here on the right in figure 6 so that we could see if it has a difference, kind of like LH2B expression, if that had a difference when you group it by high fibrosis and low fibrosis. Now our study is obviously with limitations. Our sample size is small. Our cells were upon the numbers of thousands instead of millions, and so that could have an effect with the precision of our assays. And finally, hydroxyproline is it's an amino acid that actually plays a role in this cross-linking and collagen synthesis, and so that'd be another thing to look at. Here's one final look at our poll poster. And then obviously, of course, thank you. And I've attached my information here. Please reach out, do not hesitate. I'm looking forward to your discussions. Thanks.